introduce Dr. Martha Belury in just a second, but before we do, I want to thank our co-partners. We have no sponsors for this event. Also want to thank FARI, the Fatty Acid Research Institute, for really promoting this event. Carlson Labs, Viva Omega, Nutraceutical World, Nordic Naturals, OmegaQuant, Yoli, and GoEd. These are all of our co-partners for this event that have been able to help us share information throughout. All right, let's go ahead. Dr. Belury, if you want to share your screen now, you can. I'm going to read a little bit about your bio, and I'm going to turn it over to you. So Dr. Belury, she is a professor and chair of Department of Food Science and Technology at The Ohio State University. Is the past president of the American Society for Nutrition. She conducts research in the area of precision nutrition, dietary fats, and human health. In particular, she seeks to understand how fatty acids, flavonoids, and other dietary molecules target cells to impact inflammation and energy metabolism. With her students and collaborators, she's authored over 150 peer-reviewed publications relevant to conditions such as cancer, cachexia, obesity, type 2 diabetes, psychological stress, and sarcopenia. She's also sustained 30 years of research funding from the NIH, the USDA, American Cancer Society, American Institute of Cancer Research, private industry and commodity groups. She earned her PhD in biological sciences at the University of Texas in Austin. She's also elected fellow in the American Association of Advancement in Science. In 2022, the American Oil Chemist Society awarded her with the 2022 Ralph T. Holman Lifetime Achievement Award for her work with dietary fats and health. Dr. Belury, thank you so much for being here today. Really excited about your talk.
Thank you, Tavis, and I'm so grateful too to OmegaQuant for putting together this symposium. So I actually was given the title of Demystifying Omega-6 Fatty Acids, and it's a perfect title, although I did have a slight modification, and I'm going to say I'm also hopefully going to help demythify omega-6 fatty acids, because there's a number of myths that circulate around them that I think we can clear up today, and if not, at least plant some seeds of things for you to question. Just to declare some of my funding, which could be also considered conflicts of interest, I do get funding from the Ohio Agriculture Research and Development Center, as well as I had a grant from the United Soybean Board that is no longer active, but I will be talking about some data from that study. And I also have current grants from the National Institutes of Health and the United States Department of Agriculture, as well as Pelotonia, a local funding agency. So I can't really talk about omega-6 without talking about Dr. Ralph T. Holman. Ralph, or Dr. Holman, was the person who came up with the nomenclature of omega for omega-6, omega-3, and there's actually another class called omega-9. And the reason he came up with this alternative nomenclature for chemists to use is because in mammals, humans included, of course, we cannot change between an omega-6 fatty acid to convert it to an omega-3 fatty acid or vice versa. And because we have to contain our bodies contain those types of fatty acids within their own species, that is an important thing that some types of living creatures can change. Again, we can't. And so he used this nomenclature to really help chemists understand that and follow metabolism of fatty acids. So I love the fact that Dr. Holman said life is full of surprises. I think science is also full of surprises. So what I'm talking about with omega nomenclature is omega-3, omega-6, omega-9. Most of the symposia lectures this week are going to be talking about omega-3 polyunsaturated fatty acids. And the omega nomenclature that Dr. Holman developed is referring to the fact that on this end of the molecule for this is alpha linolenic acid, the first double bond is 3 carbons. Each of these bends in the line here is symbolic of a carbon. 3 carbons in is the beginning of the first double bond for this 3 double bonded, so it's polyunsaturated fatty acid molecule. An omega-6 fatty acid, which is what I'll be talking about today, is where the first double bond is 6 carbons in from the omega end of the molecule. And there are omega-9 fatty acids, which we can make in our body. So we can make these. And the first double bond here is for this monounsaturated fat called oleic acid, where it's 9 carbons in from the omega end. So just to kind of orient you to molecules that we talk about. The 2 fatty acids that are essential in our diets, I'll be talking about today, but I'll really, again, be focusing on this omega-6 linoleic acid. So for today, I want to talk about just a little bit about what we know about linoleic acid in the diet in terms of how much we consume and what it is supposed to be doing in terms of what we understand. Then I'll show you some associative studies. And these are studies like what Dr. Harris just presented, a lot of epidemiological studies, both cross-sectional and prospective, showing the role of omega-6, especially linoleic acid, in predicting different health outcomes such as death, which is a big one, along with heart disease and other diseases, type 2 diabetes especially. Then I'm going to show you some of the research coming from my lab, where we're digging deeper into our understanding how linoleic acid impacts some of those health outcomes. And then finally, I want to discuss a little bit about distinguishing myths, beliefs, and legends that pop up in nutrition. I've seen them for over 30 years. Current one is seed oils, which we'll get to at the end of my talk. Myths, beliefs, and legends, and maybe how those arise, and then also then theories and evidence, and how theories and evidence are then to test those ideas. And we are testing it, and I think we'll show you some evidence that might counter that idea that seed oils are all bad. So first, linoleic acid is a predominant dietary polyunsaturated fatty acid that's consumed, not just in the United States, but worldwide. So it's the most dominant polyunsaturated fatty acid consumed, and it's the most dominant omega-6 fatty acid. What is linoleic acid? I showed you a molecule a few slides ago, but I'll show it to you again. This is an linoleic acid here. And this linoleic acid is esterified, meaning it's bound to a glycerol backbone, which is a 3-carbon, very simple molecule. It's an alcohol molecule that binds to fatty acids very readily. So this is linoleic acid. Up here is a saturated fat known as palmitic acid. And then on this side of this phospholipid is a phosphate group and a choline group. This is known as phosphatidylcholine. And you can see it's a linoleic acid containing phosphatidylcholine. Phosphatidylcholine is also known as lecithin, and it's an emulsifier, meaning it has an aqueous portion and a more lipid portion. And that's what all phospholipids have, is they have a more aqueous and a more lipid portion, and that brings together those two moieties that don't usually like to mix in things like cell membranes. And so phospholipids line all of your cell membranes in your cells, in your nuclei, even in the organelles within the cells. And I'll talk about that a little bit later. So linoleic acid is an omega-6, double bonded polyunsaturated fatty acid. It's an essential fatty acid, meaning it's required in the diet. It's a nutrient like any other nutrient, like vitamin A, vitamin C, vitamin E, vitamin D, all your B vitamins. It's also required. You cannot make any of this in your body. We currently have an adequate intake level for adults and for children. Adequate intake is referring to the fact that the National Academies of Sciences, Engineering, Medicine did not think we had enough evidence to actually establish an RDA. And I agree that we still don't have enough evidence to understand what the optimal amounts of these two essential nutrients, linoleic and alpha-linolenic acid, are. But currently, we think of adequate intake is between 11 grams for females up to about 17 grams for males, just to give you a sense. So grams that's about a tablespoon and a half of oil, 100% pure oil of linoleic acid. There's no such thing. So vegetable oils have mixtures of fatty acids, but just to give you a sense of the size. Alpha-linolenic acid is the only other essential fatty acid. So again, there's just two of them. This is the omega-3 essential fatty acid. You heard Dr. Harris talk about the omega index. The omega index comes from metabolites of this omega-3, and I'll show you how those form in a minute. And we need about a 10th-fold less of alpha-linolenic acid compared to linoleic acid. So you can see we need about 1 gram for females. It's 1.1 gram up to 1.6 grams for males. Common foods or common food sources for the oils that contain these fatty acids are seed oils, soybean, corn, safflower, sunflower oil. There's others too. And those are just the common ones that have been consumed for centuries. Many of these grow in different parts of the world. Safflower is more grown in arid climates. Many of you know probably soybean and corn are grown more readily in the United States and many of our regions. So I mentioned that mammals can't convert the omega-6 pathway here. You can't take one of these fatty acids and make it into an omega-3 fatty acid because we don't have the right enzymes to do that. We have enzymes that can convert this essential fatty acid, linoleic acid, called delta-6 desaturase, and this is rate limiting for all of this elongation and desaturation that can form many downstream metabolites or fatty acids that are desaturated more and elongated more, okay? Same with omega-3 fatty acids. They use the same delta-6 elongase delta-5 as the omega-6 fatty acids, but the omega-3 fatty acids can then also go on to form from this 20-carbon 5-double bonded omega-3 fatty acid other metabolites. I'm showing you one called DHA here. What's important to note, a few things. People have often wanted to express the omega-6 and the omega-3s as a ratio. I'm going to tell you now, and I'll go back to this at the end of the talk, that's outdated, antiquated idea, theory that has been proven time and again, that the low level of this conversion does not mean that if you have too much linoleic acid, you can't convert your omega-3 to its downstream products. And I'll show you evidence to support this, but this has been shown time and time again. So many of us in the lipid world are trying to get away from that ratio nomenclature because it simply pits one good fatty acid against another good fatty acid, and we know you need both. Okay, so I'm just going to put that out there, and I'm sure I'll get a lot of comments and questions about it, and I hope I do. Okay, so again, polyunsaturated fatty acids like to esterify. They have to esterify with something, otherwise they're acidic at this head group of carboxylic acid. And so they esterify, or they bind to a glycerol backbone that has a phosphorus group and then some side chain here, and that's called a phospholipid. Three big things that I want you to think about with regulating the pathways of the omega-6 and omega-3 desaturation and elongation. It is highly, highly controlled. Again, I started this work 30 years ago as a PhD student, and I was so frustrated because the theory of converting an omega-6 to its downstream product was much harder, and in fact didn't happen in vivo ever, 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 for 9 papers. And then I kept studying it and it still doesn't happen. These enzymes are very tightly controlled. I mentioned delta-6 is a rate limiting enzyme for this conversion, but each of these enzymes are very tightly controlled by the substrate that they are using and then by the product that they are making. And those products go back to feedback. That's called a feedback loop. And there's a feed forward loop as well. Okay, and that's complicated, but suffice it to say, these feedback loops and feed forward loops are very complex and depend on substrate availability, but also product availability, which people don't or usually remember when they talk about this ratio concept. Again, that I think is antiquated because of these feedback and feed forward loops. Okay, so what do people typically in the United States? Well, there is no typical diet, but if you look at a large data set of epidemiological studies, and we use the NHANES database for this, we see that generally adults in the United States have about 40% of their calories, so 39% of their calories from monounsaturated fats. So monounsaturated fats, primarily oleic acid, which is found in many meats, chicken, poultry, fish, animal foods, dairy foods, beef, etc., but also many, many baked products, processed foods, but also even some non-processed foods like things like salad dressings, which are mixtures of just a few ingredients usually. Saturated fats account for about 35% of calories. And then the rest of calories that come from fat come from polyunsaturated fats, of which about 24% is linoleic acid in the United States, and about 2% are other polyunsaturated fatty acids. So again, this is typical, but I just kind of wanted to give you the big sense of what we're doing currently in the United States. The recommendation currently is, and it's pretty general, but is to have less than 30% of your calories from saturated fat. So we know we're a little higher than that on average, and that means that these are a little lower than they should be. Okay, so that's kind of a very brief bird's eye view of some background of linoleic acid. Let's look at some associative studies and what we know about linoleic acid being important in health. So associative studies, again, are cross-sectional, looking at a relationship between, for instance, in this case, linoleic acid, and we're looking at cardiovascular disease mortality. So this is looking at just different types of or classes of fatty acids. We just look at that pie chart to show you how much MUFA. Again, we consume about 39% of our calories as MUFA. This paper looked at if you substitute for 5% of the energy from saturated fat with a monounsaturated fat, what that does to your health hazard ratio for, in this case, developing cardiovascular disease mortality, meaning dying from heart disease. What you can see is if you substitute monounsaturated fats for saturated fats, you really don't see a significant change in the hazard ratio. But if you substitute polyunsaturated fatty acids or all unsaturated fatty acids, and I think this is being driven by polyunsaturated fatty acids, you see a hazard ratio of 0.72, meaning here is your hazard ratio if you don't change your diet, at 1, and your hazard ratio for polyunsaturated fat substitution is reduced to 0.72. That's huge and that's significant. And this is hazard ratio for dying from a heart disease. Trans fats, as you can see, we've known and we continue to know that even 2% substitution of trans fats for saturated fats will greatly increase your mortality risk due to heart disease. Omega-6 fatty acids significantly reduce your hazard ratio to 0.89. Omega-3s are actually, in this particular epidemiological study, did not show an association with reducing mortality risk from heart disease. And this is just saying intake of omega-6 PUFA, and this is a clip that was pulled out from the paper, especially linoleic acid was inversely associated with mortality owing to most major causes. This was not just heart disease, but they looked at cancer and overall mortality. This same paper then drew up a model to suggest for every percent increment increase in trans fats, what you would expect in total mortality. And you can see trans fats greatly increase our risk for mortality from all causes. Saturated fats significantly do as well, but not to the steep extent that trans fats do. Monounsaturated fats decrease our risk, and then polyunsaturated fats decrease our risk even more. So this paper was published in 2016. And then there was a more recent paper published in 2020 looking again at all-cause mortality and then heart disease, cancer, et cetera. And this paper was a pooled meta-analysis of several prospective studies. In this paper, the authors looked at the dietary intake of linoleic acid and its association and relative risk to, in this case, death due to all causes. And what they found is that relative risk was 0.87, which is significantly lower risk for mortality when you have higher amounts of linoleic acid in your diet. Okay. Similarly, they then looked at cardiovascular disease mortality, induced mortality, and they see about the same relative risk, 0.87. So both of those tables are looking at relative risk and associating that with higher linoleic acid is reducing your risk for both total all-cause mortality as well as cardiovascular disease mortality. They then went on to look at biomarkers of linoleic acid. So here's adipose tissue levels of linoleic acid, phospholipids from the blood, cholesterol esters from the blood, et cetera. And using any of these tissue types and pooling them with a lot of adjustments to the data for things like location, different sites around the world that were used in these studies, what they found is a relative risk of 0.91. Again, it's showing us that higher levels of linoleic acid that are in your tissues, which have to be coming from your diet since you can't make linoleic acid, higher levels are associated with reduced all-cause mortality And the same is true for cardiovascular disease mortality, which is about 0.89. So these are just two epidemiological papers that show us that there could be some benefit to omega-6 fatty acid intake, especially linoleic acid in your diet. In fact, for over 70 years now, the American Heart Association has recognized that omega-6 fatty acid, especially vegetable oil-derived linoleic acid, could be beneficial for heart disease. This is a clip out of this advisory paper from the American Heart Association saying polyunsaturated vegetable oil reduced cardiovascular disease by 30%, similar to the reduction achieved by statin treatment. Now, if you know anything about heart disease treatment in the United States, most people go onto statins if they have dyslipidemia, meaning high cholesterol, LDL, or total cholesterol, or low HDL. Sometimes they're put on statins too, but mostly it's for high cholesterol, LDL. And we're looking at something that's a nutrient that could have the same benefit. But I'll just let that lie and let you think about that for a bit. Recently, and I would say this is really more in the last 10 years, although when I dug deeper, it was being studied in Europe decades earlier. People have started to show that not only is linoleic acid associated with reduced risk for heart disease, but it might be associated with reduced risk for type 2 diabetes. So heart disease and type 2 diabetes are some of the most common chronic diseases in the United States. Type 2 diabetes is associated with central obesity. It does actually lead to heart disease in many patients, and it also has a genetic component. But we also know that diet is important in type 2 diabetes risk and management. So this paper looked at omega-6 fatty acid biomarker and the development or incidence of type 2 diabetes. It pooled analysis from 20 different cohort studies all over the world. So again, the source of the dietary linoleic acid didn't matter. It was pulled from many, many different countries. And what you can see here is that for each quintile of an increase in linoleic acid in the body, so biomarkers of linoleic acid, there was a significant decrease in relative risk of type 2 diabetes. The data, like data from the past few correlative studies that I showed you, were adjusted for many things. In this particular study, age, BMI, sex, race, omega-3, polyunsaturated fatty acid levels, aspirin use, and polymorphism in this fatty acid desaturase gene, FADS1. And so what this is telling us is that higher levels of linoleic acid are associated with reduced risk for type 2 diabetes. Could there be a causative link? And of course, an associative study can't prove causation, but it definitely suggests something's going on. This is one of many studies now showing that there is a very significant relationship between linoleic acid levels in the blood or in tissues and in the diet with reduced risk for type 2 diabetes. And what that's about, even when we adjust for things like BMI, sex, and race, and age, which are very high risk factors for diabetes, you get higher risk as you get older, unfortunately. We don't know. So we're testing for that. What's really important here too is if you look at this other omega-6 fatty acid, arachidonic acid, which is formed from desaturation and elongation of linoleic acid, there's no correlation of higher risk or lower risk. It's pretty much mute. Okay, so moving on to the third part of my talk. What do we know from studies in my lab? I showed you this pathway of the omega-6 and omega-3 metabolism to linoleic acid down to arachidonic acid, alpha-linolenic acid down to EPA and DHA. I wanted to see whether if we increase dietary fortification of linoleic acid, if that would indeed change some of the downstream metabolites, and also if that would change the eicosanoids or the prostaglandins, these inflammatory modulators that form from the fatty acids. So we conducted a very simple trial. It was a single-arm intervention where we fortified women's diets with about a teaspoon a day of linoleic acid-rich oil. We recruited 15 postmenopausal women who had metabolic syndrome. We call this the Healthy Oils for Women study. And then we measured blood levels of linoleic acid and other fatty acids throughout the 16 weeks. We also, at week 0 and 16, measured prostaglandins, leukotrienes, and some of these metabolites of fatty acids that we know can affect vascularization, vasodilation, inflammation in the body acutely. Now, this is a very busy slide, okay, and I apologize for that, but it's called a network analysis. So a couple of slides ago, I showed you this very simple pathway. Now, what we know is that simple pathway has gotten a lot more complex now that we have better instrumentation for measuring fatty acids and their metabolites. Dr. Harris mentioned gas chromatography, which he and I and others still use to measure fatty acid composition of the blood. But in this study, we used HPLC mass spec to detect all of the fatty acids and all of these metabolites, many of which are associated with pro or anti-inflammatory or even resolving effects in the body for inflammation. And so what we see here, and this is the molecule of linoleic acid that we fortified in the women's diets here 16 weeks. We see no increase or decrease. If there were an increase, you'd see a red dot. If there were a decrease, you'd see a blue dot. The size of the dot tells you how much of the change, either increase here or decrease here, that we would see from fortifying after 16 weeks. You see no increase of linoleic acid. You see no increase of its downstream metabolite, arachidonic acid. You do see many of these lipoxygenase and epoxygenase products from linoleic acid. Many of them are actually associated with pro-resolving or other effects in cells. So I'll go through some of the effects that we study, but these are not thought to be pro-inflammatory, meaning promoting inflammation in the body, at least at this time. So we see many of those increase, which is what we had expected. We did not expect to see this omega-6 metabolite decrease, because people say if you increase linoleic acid, you're going to increase all the omega-6 metabolites. Again, we don't see a lot of these things change at all. We even see a decrease of one. But I want you to note what's even more puzzling here is we actually see some omega-3 metabolites increase when we fortify diets with omega-6. Why is that? Let's go back a minute. These enzymes, as I mentioned, have feed forward and feed backward regulation, meaning some of the substrates that go on to be formed from linoleic acid shut down or turn up some of these enzymes. And it depends on so many things in the cells. So many things in the cells, like what the cell needs at that time, at that very microsecond that it's doing all this metabolism, what the membrane needs, what's going on energetically in the cell, et cetera. And again, that's stuff we study. It's very complicated, but the fact that you see increases in the omega-3 metabolites tells you one thing that you can understand. It is complicated. It is not as simple. If you increase omega-6, you're going to decrease omega-3, which in fact we don't see here. So I just want you to note that. One of the other things we know is that, go back here, some of these metabolites, these, let's see, 13 HOD here, 9 HOD here, and some of their other metabolites actually bind to molecular receptors in cells and change gene expression. And I've studied this for years in my lab due to several great collaborators that have taught me many things about how to do this and work with us to do it. And one of the things we know these HODs do is they bind to this receptor called PPAR gamma, which sits in the promoter region of certain genes. One of the genes that PPAR gamma can turn on is this protein, this level is mRNA level of adiponectin, which then gets translated into protein, which gets in your blood. What's important is adiponectin is helpful for reducing diabetes and atherosclerosis events. Okay, so it's a protective protein. What we see, we found this now in 2 studies, is that linoleic acid fortification, even after just 4 weeks, increases this adiponectin by 4 weeks and maintains it throughout the period of fortification when our women were on the linoleic acid rich oil. Okay, so this isn't what we would have expected. We didn't expect to find it, but this is what we found. And so moving forward, I want to show you another study where we measured how linoleic acid could affect women with type 2 diabetes. So this was a double-blinded, parallel arm crossover, meaning women were on either a dietary supplement called CLA or tonalin for 16 weeks, or they were on the linoleic acid-rich safflower oil for 16 weeks. We did a number of measurements with our women at each of our 4-week visits, exchanged supplements. In this case, we used pills for their supplements so we could help them adhere to the protocol. Then after 16 weeks, we pulled the women off of their supplements, allowed them to rest for 4 weeks, and then they went on to the other treatment. Again, this was a randomized controlled study, so we didn't know what treatments the women were on during the phases of the diet periods, et cetera. What we found is that the linoleic acid-rich safflower oil reduced fasting glucose, which is a primary marker that most people with diabetes measure fasting glucose when they get up in the morning. It helps them gauge what they're going to have for breakfast and what insulin they're going to need if they're using insulin. It also helps them understand if they're doing something with their diet or exercise that they might need to change, right? So we saw fasting glucose between week 0 and 16 weeks was reduced by about 16 milligrams per deciliter, which is significant. The women, we were asking them not to lose weight, not to start a significant exercise during the study. If they did, they just informed us and we accounted for that in the study, but nobody really did change their diet or exercise from what we measured with our 3-day diet and activity records. The dietary supplement that's out for weight loss right now called tonalin had no effect on fasting glucose. The other thing we saw was that linoleic acid fortification was able to also reduce HbA1c, which is a long-term measure of glucose control. And it was reduced by about 0.7%, which is unit percent of HbA1c. So these were very exciting to us, somewhat puzzling, because when we published this in 2009, there weren't a lot of studies looking at linoleic acid and its role in diabetes. We also measured body composition using DEXA in the women at the beginning and end of each diet period. So at week 0 and 16, and then again for their second diet period, week 0 and 16. And surprisingly, we found that linoleic acid oil fortification increased lean mass measured by DEXA, and CLA had no effect. Linoleic acid also reduced trunk adipose by about 3.7 pounds of trunk adipose in the 16-week period, and the dietary supplement that is thought to be a weight loss supplement had no effect. Finally, we looked at some markers of inflammation, CRP and IL-6. CRP was actually significantly reduced in the safflower linoleic acid-rich supplemented time period, and this adiponectin again was increased. So when you see the same thing in more than one cohort study, you start to think there might be something real, that it's not just some anomaly of how the study was done or what cohort we were studying. Looking back, when we looked at that correlational study showing that every quintile of linoleic acid in the blood was associated with reduced risk for diabetes, what we're showing in our women's diabetes study is that if we fortified their diets with less than a serving size of linoleic acid-rich oil, so a teaspoon and 2/3 approximately was what they were receiving each day, we were able to reduce their fasting glucose, reduce their trunk adipose mass, and reduce CRP and several other measures. So this is just kind of showing you that 10% change in the blood of these women in our diabetes study was able to be associated with the same effect that we see in an associative study. Okay, so let's move on to mechanism, and then I want to get to what this means in terms of myth, science, or whatever in the middle. Okay, my lab looks at how polyunsaturated fatty acids get into phospholipids. So I mentioned that polyunsaturated fatty acids, like linoleic acid, like to bind to this glycerol. In this case, again, is this phosphatidylcholine, also known as lecithin. In the mitochondria, there's a certain type of phospholipid that is unique only in the inner mitochondrial membrane. And this is important because this inner mitochondrial membrane phospholipid is known as cardiolipin. Many of you probably haven't heard of it. I hadn't heard of it until I started studying it. But again, it's in the inner mitochondrial membrane where the electron transport chain occurs. So if you remember that the mitochondria is the organelle in all of your cells that generates ATP energy, it happens by the electron transport chain, which is sitting on this inner mitochondria membrane. Importantly, this inner mitochondrial membrane prefers one fatty acid, and it's linoleic acid. There are many reasons that scientists speculate why. It could be that it only has two double bonds, but it has enough curvature in its molecule to support cristae formation that can rapidly increase and decrease according to energetic needs. It could also be that it pulls the electron transport chain machinery together better, meaning all of these proteins have to be pulled together for electrons to be transported from one protein to the next with these molecules, NADH and FADH2, okay, to allow for ATP synthesis here. And so for some reason, linoleic acid-rich cardiolipin, and it's probably somewhat related to its three-dimensional stoichiometric interaction with these proteins, is the unique best option for cardiolipin molecules to support energy production. So two students of mine wanted to study this in mice. And so we decided to do a study comparing in mice, healthy mice, a linoleic acid-rich diet or a lard diet, which is basically a mixture of saturated, monounsaturated, and polyunsaturated. So lard diet, believe it or not, is fatty acid-wise a lot like what the typical American diet is that I showed you in that pie chart. And I want you to just focus on this right graph. The left-hand graph is just looking at forelimb grip strength in mice. So we actually have them do little chin-ups, literally. But we also measure hind limb grip strength. And we found that hind limbs were significantly stronger in the mice fortified with the linoleic acid oil. That was a very unique finding and kind of matches what we found in the women's diabetes study with increased lean mass. We collaborate with a fabulous chemist, physicist, named Dr. Genevieve Sparagna, and she can do targeted lipidomics of muscle mitochondria using this HPLC mass spec Molotov system. So we isolated the muscles of the mice after necropsy or during necropsy, isolated the mitochondria and the lipids from those mitochondria and sent them to Genevieve, who's in Colorado, and she analyzed the lipids under blinded conditions, because we do most of our work under blinded conditions. In other words, not knowing treatment groups. And what she found is that, like we expected, the linoleic acid-rich oil-fed mice, which is the gray, increases linoleic acid-rich cardiolipin, which we call tetraniliol, meaning 4-linoleic acid cardiolipin, or L4CL. So linoleic acid diet increases this cardiolipin species, which is rich and we think optimal for electron transport. And the control oil, which again was lard and contained oleic acid, actually increases oleic acid-rich cardiolipin. And that was, this oleic acid-rich cardiolipin was reduced in the linoleic acid-fed mice. And you can see some of the other species changes. So we wanted to measure whether this would have an impact on mitochondrial activity. And you can see the linoleic acid diet had significantly higher mitochondrial respiration activity compared to the lard oil diet. And we measured this in muscle, but we also measured it in other mitochondria-rich tissues, including brown adipose tissue. And we saw similar findings. So we're following up on this currently. But to me, this all suggests that linoleic acid could be important in mitochondria, and whether that has an effect then to go back to affect type 2 diabetes is really one of the questions we have. We like to translate our preclinical mouse work to clinical outcomes, because early on in my doctorate, I found many things in mice that didn't pan out when we tried to either test it in humans or when we looked at the epidemiological studies to see if there was a way to translate our work. And so one of our, two of my lab people, Dr. Rachel Cole and Dr. Austin Angelotti, conducted a very simple 2-week parallel arm study. We call it the Healthy Cookie Study. where healthy adults were given cookies that were that had 12 grams of oil in them, either the oleic acid rich oil cookie, which was mostly oleic acid, and the linoleic acid rich cookie, which was mostly linoleic acid. This is grams per serving. We asked the participants to consume one cookie a day. So just add it or use it as their dessert or part of their snack for 2 weeks. And what we found, again, is in people, we could increase this linoleic acid rich cardiolipin, which again, we think is important in mitochondria. And our newest study is funded by the NIH to look at how linoleic acid enriched cardiolipin can then be translated, hopefully, and maybe it won't be, and that's the beauty of science is we don't know until we test it, but see if this linoleic acid rich cardiolipin can support and sustain higher metabolic mitochondria respiration. And so that's currently an ongoing study. But this healthy cookie study gave us the data to suggest, wow, we can make a change in people, which this is, to my knowledge, the first study that's ever shown this, that linoleic acid increases cardiolipin that's rich in linoleic acid in people. So currently we're pursuing a couple of mechanisms of how linoleic acid could be improving glycemia in the women with diabetes. I mentioned we know that linoleic acid increases adiponectin in both metabolic syndrome women as well as type 2 diabetes women, reduces trunk fat, increases lean mass. We think part of that might be through this cardiolipin, to this tetralinoleal cardiolipin species that increases the electron transport chain. And that would improve glycemia because part of what happens in diabetes is if your glucose can't get into your muscles, other things start to get dysmetabolized. In other words, not metabolized properly, and your muscles can become fatty. You can increase your body fatness, adipose tissue mass, because higher insulin levels in your blood do that. That's part of what insulin does naturally. And so we think this is a very important part of how linoleic acid could be improving glycemia. We're also still very interested in understanding how linoleic acid's metabolites, like these 9 and 13 HODs, can act as agonists for PPAR gamma to increase proteins like adiponectin, which are known to improve glycemia. We'd like to show that this is all happening in people. We've shown parts of it, but we're still hoping to connect it all together. So last but not least, there's a lot about omega-6 fatty acids that I've heard over the years. The latest one is the seed oil theory. I don't know what it's called, but I want to talk about what happens with a lot of oils in the United States first. So a lot of oils get hydrogenated to make oils, which are liquid at room temperature, to become solid at room temperature. So this is the oleic acid molecule. This is a monounsaturated fatty acid. Hydrogenating means you're adding 2 hydrogens to this double bonded carbon oil so that you make it saturated. So there's no more double bond there. And that actually causes the molecule to be more solid. The crystalline, the fatty acid crystallizes at a higher temperature. Okay. So meaning it's more solid at room temperature. That's what stabilizes a lot of fats for different food products, whether it's baked products, other food products. It is what it is. It's hydrogenation. You can see this is a hydrogenated product food label where vegetable oil is the first ingredient, but it says vegetable oil blend, partially hydrogenated. What happens with partial hydrogenation is that you also get the formation of these double bonds accidentally, and they're very low, but they do happen, called trans fats. So over the years, vegetable oils have become a little different in our food supply. I'm going to jump to that. This is the label of what I call the shifting identity of vegetable oils in the United States. And this might have, this doesn't have any relevance to the seed oil controversy. That's a whole other thing that I want to get to. This has to do with the fact that our oils used to be naturally high in linoleic acid, and that was polyunsaturated fatty acid, because that's what most vegetable oils have in them for their poly content, and low in mono. And over the years, many crops have been changed to be higher in monounsaturated fatty acid and lower in poly. Now, the reason is I'm going to go backwards for a minute. if you have one double bond that you have to hydrogenate to make it solid, you have a lower chance of making this trans fats than if you have two double bonds, which is what a polyunsaturated oil has in it. Okay. So all this to say that what we used to know were high in linoleic acid is actually no longer necessarily true. Many of these seed oils are now high in oleic acid. And I don't think there's any bad thing this is not a bad thing to have more oleic acid rich oils. In fact, they're more shelf stable. Many people don't use their vegetable oils in a timely fashion. And if you have a shelf stable oil, it's not oxidized on your shelf. If you do smell anything like fishy oil, you should throw it out. It means it's oxidized. But this is also important for our food supply because we know just a tiny amount of trans fats has a huge effect on cardiovascular. Oh, no, sorry. This is all-cause mortality in people. And we know for cardiovascular disease mortality, it's the same. Trans fats, tiny amounts in your body are not good, period. So that's something we absolutely want to continue to help people eliminate is trans fats. I'm going to jump through and go to this. Seed oils. So what's the deal? I actually was just literally surprised when somebody said, what about seed oils? What do you think about seed oils? You know, as a dietitian and then a nutrition professor, I get asked everywhere I go, what do you think about blah, blah, blah? Most of the time, I had no idea what they're talking about. But seed oils, because I study oils that do come from seeds, I thought I better find out. Parts of the controversy may be about the omega-6 content, but I think a lot of, and I haven't looked at a lot, okay, the few times I've Googled this to look at influencers, some of them are saying it's not the omega-6 content, it's the fact that their oils are extracted and not pressed. And that means that they don't have any vitamin E left or other antioxidants. In fact, they've had oxidation added to the oils, which will then promote oxidation in people's bodies. And by oxidation, I'm talking about non-enzymatic oxidation. These are things that that could be increasing free radical generation, okay, and oxidative stress. So I have another study that we're just still getting ready to publish. And so this is very, very hot off the press. But we did a study in people with non-alcoholic fatty liver disease, where they were supplemented with 3 products a day, delivering about 22 grams, 25 grams of fat a day, oil a day, either as linoleic acid rich soybean oil or control oil, which was lard, again, mimicking the typical American diet fatty acid composition. And our primary outcome was to look at liver fat, thinking that if people with non-alcoholic fatty liver disease could reduce their fat with this polyunsaturated fatty acid, this could be useful for them and beneficial as part of their dietary approach to managing NAFLD. But one of the things I'll show you the primary data just because it's very exciting. So we measured liver fat using magnetic resonance imaging. And what we did find is that the soybean oil did significantly cause a decrease in fat of the liver in comparison to the control group, which had no effect. So the treatment effects are different. You can see both of the deltas within each diet group are not different. So the effect was different between the groups, but we didn't see a significant decrease. So I want you to pay attention to that. We're still going to, I can address that in a couple minutes with what we think is happening. Here's what I want you to know about going back to the seed oil. Soybean is one of the seed oils that the influencers on the internet are trying to tell you not to eat. When we looked at oxidized LDL in people with non-alcoholic fatty liver disease, there was no difference, no change and no difference between the groups. Okay. This is lard, by the way, that at least one politician now says you should start eating. It actually has no effect on oxidized LDL, but neither does soybean oil. We also looked at the binding protein for, can't remember it's a polysaccharide that actually induces inflammation. So if you consume some of this, it induces inflammation. We saw that the binding protein, which should go up if there's increased inflammation, again, didn't budge with our soybean oil. So just want you to think about some of the findings and think about that in the relevance to what we're talking about with seed oils. I want to wrap up just by saying, for demystifying omega-6 fatty acids, think about the science versus the belief. And remember that in nutrition, there's a lot of influencers who want to sell a book or sell a supplement or sell something that may not be using first-based deep science and evidence. And dig deep. If you need to dig deep, contact a scientist at your local university. But don't take everything at its word, because there's a lot of influencer out there that is not using science first. I would say seek to boost, and I think this will be maybe coming up throughout the week, seek to boost both your omega-6 and your omega-3 fats in your diet. We know that that simple parallel pathway competition theory that people had many years ago, in fact, about 40 years ago, actually isn't quite right. So please question articles and internet information that still uses this ratio, because we think both are important for heart disease prevention, type 2 diabetes prevention, and other chronic disease prevention, and hopefully reducing your risk for early death. There's a lot we still need to know about elucidating cellular mechanisms of fatty acids. The answers aren't quite there yet. And I know people get frustrated that nutrition is, why don't we have all the answers to nutrition? Well, it's probably the same reason we don't have all the answers to heart disease pathogenesis and why some get it, some people don't, or cancer, et cetera. Same as evidence for setting RDAs for linoleic acid and alpha-linolenic acid. I hope before my days are over as a scientist, I can see the establishment of these RDAs. And for that, we need more fundamental research in nutrition funded by the NIH and other organizations. And with that, I'll close with my acknowledgements to all of the fabulous scientists who collaborate with my group, many at Ohio State, but many around the world, and my fabulous lab group here of my graduate students and staff scientists, and including some of my scientists who aren't pictured here. I'll close.
Excellent. Dr. Belury, thank you so much. Just incredible information. And hopefully that helped answer a lot of people's questions around omega-6 fats, AKA seed oils. All right, let's get to the Q&A. So there's a good bit of questions we want to go over. With how many seed oils we have in the standard American diet, how does recommending additional linoleic acid supplementation make any sense?
Good question. So RDAs were generally for adults set based on your age. If we look at RDAs for children, they're based on body weight. If we know that people are generally getting larger in the United States, one of the questions that some people have about what we need for nutrients that are essential, like linoleic acid and alpha-linolenic acid, are maybe if you have a larger body, you might need a little more, kind of like what we do with children's RDAs. But all of that is to be determined. Whether we get enough linoleic acid is, again, we can't say whether we do or not when we don't have an RDA. And that's kind of the conundrum that nutrition research is in, not just with fatty acids, but with a lot of our fundamental research that we still don't have worked out yet.
Are there known typical levels? I guess it says of fatty acids, but I'm not sure if they're known, if they met linoleic acids, intake for vegetarians and vegans.
There are many studies showing linoleic acid and alpha-linolenic acid are generally higher. Not always, of course, and it depends, because vegans can have a lot of saturated fats and other things that influence their fatty acid composition. So yes, there are papers that look at that. We haven't looked at how vegans would respond to fortification with a modest amount of linoleic acid or other. We also test omega-3 fatty acids in my lab for other things, but that wasn't my role to report that today.
How much linoleic acid per day do you feel is really the optimum level, and how much is too much?
Yeah, absolutely. For everything in our diets, everything in moderation. I know that's a very boring message that all the dietitians that you'll ever meet will say. Looking at what you do and what your body does to metabolize linoleic acid or any fatty acid is important, because we all have some genetic propensities that could change how fast you metabolize a fatty acid or how long you keep one of its metabolites. So that could go back to influence what we think we need. So again, I mentioned the adequate intake right now is between 11 grams and 17 grams for most adults, most age segments in the adult population. But that's adequate intake. And whether that's for optimal heart disease prevention and diabetes prevention, that's a whole nother kind of step that we have to take when and if we do that for setting our DAs. So right now it's 11 to 17 grams a day, which means about 20 to 30 grams maybe of oils that are rich in both of the essential fatty acids. But you generally get those in a lot of foods you eat. Whether some people aren't getting that or not, we have shown that as people get older, they tend to have less in their diets. But Dr. Harris has actually shown as people get older, they definitely have less linoleic acid in their erythrocytes. So what that's about, that's a whole nother story. But reaching an optimal blood level might take more for some people than others.
Which is more important in determining plasma linoleic acid levels? Is it LA intake or mutations in the fatty acid desaturase genes?
Yes, and both. Um, Well, great question. So some people have mutations in this desaturase, fatty acid desaturase, either 5 or 6. Either one will promote more rapid metabolism of linoleic acid and alpha linoleic acid to its downstream metabolites. So a very good question that I think I want to address at some point is whether people who are faster metabolizers might actually be at a higher propensity for some muscle disorders, including heart failure, which is a muscle, heart is a muscle, as well as muscle loss, which is one of the studies we're doing right now in people with sarcopenia.
Excellent. Any thought on free range beef tallow used instead of vegetable oils? I guess they mean for cooking.
Yeah, well, free-range beef tallow will be very high in monounsaturates and saturated fats first, so the polys won't be very high. I'm concerned about that. I don't think that beef tallow or lard is something we want to go back to. I think the people who are promoting that might not study how oils work in the body, and I'm concerned that again, there's a lot of influencer messaging coming out on that I'm not sure those people are reading the scientific evidence for. So I don't support that we should go back to beef tallow. It can be useful and tasty for some recipes, again, in moderation. There's nothing wrong with any of this, but I don't think that should be our day-to-day cooking oil.
Do excess intake levels of other fats, such as trans or saturated, have an inhibitory effect on the mitochondrial benefits of linoleic acid?
In fact, we don't know about the trans fats that form from partial hydrogenation, but we do know that saturated fats, if those fatty acids get in the cardiolipin, they absolutely cause electron leakage and poor mitochondria function. So that's kind of the point of fortifying with linoleic acid to get it in the cardiolipin, is to keep that cardiolipin healthy and not allow for it to be depleted of linoleic acid and pick up whatever it can find, kind of scavenging whatever fatty acids are in the area.
Given this information, what would be an appropriate placebo to use when studying omega-3s instead of omega-6 rich oils like corn oil?
That is such a great question. That is something that keeps me up many nights. What is the proper placebo or control fat or oil? For different studies, we've used different controls depending on our research question, right? And so if we want to compare our newest study is looking at high oleic acid safflower versus high linoleic acid safflower. So, and the reason we've chosen that is we think they're most similar except for fatty acid, right? One fatty acid. But sometimes you want to use a controller placebo that's more like what people are already doing. And that's why we chose lard in the fatty liver study, because lard, as I said, is 35% mono, 35% saturated, and then about 30% polyunsaturated. So it's a problem too. And then to be able to compare studies is then a whole nother thing, right? You need to have the same control to know what your baseline is. Whoever asked that question, I'd love to talk with you more. I mean, we've even designed oils that are mixtures and put them in pills. That's a nightmare because nobody wants to handle the oils just because it's so complicated. But yeah, good question.
Question then as a personal part to this question, someone's asking, given the change in crops to produce more monounsaturated fats versus polyunsaturated fats, do you still recommend using those oils in cooking at home? What is your go-to oil for cooking?
I do look at, because I'm a dietitian, I'm a dweeb, I show those photos of labels. I actually look at labels. If oils will list the poly, which they're not required to, they're only required to list saturated fat and total fat. But if they do list poly and it's higher, poly is more. So I remind my family of this daily almost. Poly is more, and you want more poly than mono, just to know that you're getting more of that polyunsaturated fatty acid, linoleic acid. There are still many commodity, not commodity, what am I saying, generic oils. So your store brands that are vegetable oils, those generally are still higher in linoleic acid according to their label, fatty acid content if they show polys. There are also some oils that are not mass produced for oils. I know it's on the seed oils, Hateful Eight or whatever they call it, but grapeseed oil currently still has higher linoleic acid, and it's very high in linoleic acid. Safflower oil is in Ohio, it's not very usually found to be high in linoleic acid, and I think regionally different grocery stores are sourcing different oil companies for their oils.
Out of curiosity, what's your opinion on avocado oil, especially with being able to handle some of the higher temperature points?
Yeah, actually, the mono all of the vegetable oils that I've talked about, so safflower, not sunflower, safflower, and grapeseed oil especially have very high smoky points too. But avocado oil has some unique taste that some people like. It has also, like olive oil, has a lot of polyphenols, right? And those are a whole nother group of compounds that could have health benefits. Again, we don't know a lot about them, but we're learning more about polyphenols and those antioxidants and how those work in the body.
Just a couple more questions. I know we're kind of at the middle of that hour, but we don't have a session after this, but I want to be respectful of your time. Any concern with consuming refined seed oils?
No, I think if I had a concern about refined seed oils, I don't see that it has any bearing on what I'm seeing in the epidemiological literature or what I'm seeing from other people's labs looking at randomized controlled trials. I just showed our randomized controlled trials with linoleic acid rich seed oils. But I think if we were to find something, we would have seen like an increase in oxidized LVL or an increase in LBP or an increase in some of our other markers of oxidation. So I'm not seeing that the evidence is there that refined oils are any different than refined flour or refined rice or anything that's processed so that we can get more to the access of the nutrients in them. But I'm just not seeing the evidence yet.
Do you think having a target linoleic acid level in red blood cell membranes, like an LA index, like having its own index, like omega-3, would be useful to test? Would be a useful test to help people optimize their LA intakes?
Yes. And if that person, whoever asked that question, can form an algorithm to determine that. I think Bill and I kind of talked about it a bit, Bill Harris and I, but I think it's an important thing to consider. For 70 years, we have known that linoleic acid rich oils lower LDL cholesterol. That's for 70 years. And that's not changed over the 70 years. We still see that. So we know that there's some benefit, even if that doesn't always translate to lower heart disease risk. I think there's something to these epi studies that are showing lower cardiovascular, cancer, and all-cause mortalities and disease risk.
Last two questions. It would be great to understand, and it's not a question, it seems like it's a statement, but maybe there's a question from this. It would be great to understand genetic mutations such as FADS2 and how fatty acids are metabolized. I have mutations on 26 FADS2.
Oh, wow. I agree. I just wrote a grant to the NIH about that. So hopefully we'll get it funded. Contact me. Maybe we can get you enrolled in this study.
I just want to share my screen real quick before we answer the last question, because I want to give a little highlight to the Fatty Acid Research Institute and the work that Dr. Harris and his colleagues and yourself are doing. So if you have any questions, if you'd like to support FARI, you can click on that link right there, or scan that barcode and donate to support more fatty acid research. Last question. What's your message to the noisy fringe demonizing the omega-6 fatty acids or seed oils? What message would you like to give those people?
Well, I didn't show the last slide of my talk, but it's a grizzly bear eating a salmon that's swimming upstream. And often I feel like in nutrition and in food science, we handle a lot of fringe influencers that might want, I don't know if they want attention or if they want to sell something. I would always just be wary of anything that's promoted as research when the person might not have done the research other than reading research, which is not doing research, it's reading research. So contact dietitians. If you're a dietitian and you don't know where to go, contact your local universities for nutrition researchers. I'm always happy to talk about fatty acids because it's basically all I study. But yeah, it's a constant challenge in nutrition is to handle the kind of fringe messages that go out there.
Great. And we'll share, I don't know if you want to share your email, if you shared that before. If not, we can, if other people have questions, questions that are not answered, we'll get you connected with Dr. Belury. That way you can reach out and have some questions. I know you said you had some people that you're like, hey, the question on placebo, reach out. So definitely, she's with Ohio State. That's all I'll tell you. You can probably find her pretty quickly if you use her name and type that information. Okay. Doc, thank you so much for the time. Phenomenal talk. Great job all around.